This is Sonika from Botany Department of Arya College, Ludhiana. Today's topic is linkage and crossing over. The number of genes in an organism far exceeds the number of chromosomes, which is very true. Gene tally for Daphnia pulex is around 31,000 genes, which is stacked up against other organisms like humans have about 23,000 genes, close in number to mice, roundworms, and a small flowering plant called Arabidopsis thaliana, on which a huge amount of research has already been done. Fruit flies have about 14,000 genes, yeast about 6,000, and E. coli, which is a bacteria, it has about 3,200, and zebrafish has more than 5,000 genes. So, a single chromosome bears several genes. Linked genes are the genes which are present at the same locus, that is, the same position, which have the tendency to be linked together from one generation to the other and are not disturbed by the meiotic recombination. So, linkage is defined as the tendency of genes to remain together during the process of inheritance, means from one generation to the next generation. When two loci were linked genetically on the same chromosome, they did not segregate. So, the extent of linkage is the closer the genes, the stronger the linkage, and vice versa. Linkage of genes in this linear fashion is in the chromosomes. Linkage. The genes that show linkage are located on the same chromosome. A linkage group is formed by all the linked genes in a chromosome. The strength of the linkage between two genes is directly proportional to the distance between them, means the more the genes are farther apart, the less there will be a tendency of occurring the case of linkage. Then comes the type of linkage. Number one is complete linkage, when two or more genes tend to remain together on the same chromosome and are inherited from one generation to another generation for many generations, and this phenomenon is known as complete linkage. It happens when the chromosomes do not undergo any breakage by accident during gametogenesis. Next is the incomplete linkage, when the linked genes tend to separate on some occasions during the course of inheritance. So, this phenomenon is known as incomplete linkage. It occurs during the process of crossing over during gametogenesis. The genes present on the homologous part of sex chromosomes exhibit the phenomenon of incomplete linkage. Then comes autosomal and sex linkage. Autosomal linkage, which occurs in the linked genes that are present on the autosomes. Sex linkage occurs in the linked genes that are present on the sex chromosomes. Linkage groups in fish detected by Wing, that is in 1923, for example, the XVI and XEL genes in the guppy are linked on the X chromosome. Although both genes may cross over to the Y chromosome, it is assumed that they located only on the X chromosome. Then comes the crossing over. It is a physical exchange of parts of two non-cystochromatids of homologous chromosomes following synapses at meiosis. It results in recombination of genes, means it will result in recombinance, means the progeny will be a new one, a recombinant one, a variant one. So, one or two or more fragments may be interchanged during crossing over. So, here is a basic diagram of crossing over process in which chromosome A and chromosome B is shown, and the second diagram is showing the crossing over process in which you can see the genes that are represented by E, F, G, H, I in the caps and the lowercase letters are being swapped. So you can see over here on the first homologous chromosome, the chromosome A, where the small h or the lowercase h was present, here it has been replaced with the capital H gene. So, the frequency of crossing over of a given pair of genes is not constant. It occurs from organism to organism. For example, crossing over frequencies are higher in the females than in the males. And other factors are temperature, nutrition, sex, age also influences. It is a part of meiotic mechanism during gametogenesis and brings about variation and leads to evolution through natural selection. Now, the single crossover or the single crossing over is in which only one chromatid of each chromosome is involved. So, the single crossover is of the most frequent occurrence. It occurs very frequently. Then comes the double crossing over, in which both the chiasmata is the place where the crossing over takes place, maybe between the same chromatids or between the different chromatids. Two or three or all of the four chromatids of the homologous pairs of chromosomes are involved in the process of double crossing over. So double crossover gametes are produced. This is of less frequent occurrence as compared to the single crossing over. So here is the picture of crossing over, specifically the double crossover, in which the chromosome 1, which is shown in red, and chromosome 2, which is shown in blue. The crossing over or the double crossover is shown between the non-sister chromatids of these two homologous chromosomes at two places, means two chiasmata has been formed, or the two crossover sites. Multiple crossing over is that more than two chiasmata are formed. Corresponding to the number of chiasmata formed, it is called triple if the three chiasmata are involved, quadruple if four are involved, and so on. Multiple crossing over does not occur frequently, it is very rare. So the significance is that it provides a direct evidence of the linear arrangement of genes in the chromosomes. Chromosomes map can be constructed. It gives rise to new combination of genes and hence variations in the offsprings. Chromosome mappings, which means the student constructed first chromosome mapping, means the location of the genes on a chromosome. So the map showing the position of genes on the X chromosome, it was first constructed by Sturtevant. Genetic maps of the chromosomes are also known as chromosome maps. Gene mapping refers to the analysis of loci or the position on the genome, revealing the linear order of different genes on the chromosomes. There are two types of gene maps, physical maps and genetic maps. Physical maps are based on the assignment of loci to chromosomes. It is accomplished by the methods of somatic cell hybrid panels, in situ hybridization, and comparative mapping. In physical maps, the coordinates are the chromosome regions or bands. The distance between the two loci are measured in kilobases. The units are kilobases. Genetic maps are constructed by studying the meiotic recombination between two or more loci through linkage analysis. A new locus are assigned to a chromosome following the inheritance of the new locus and of an already mapped locus. So it do not provide absolute location of loci, but they reveal the genetic distance of the loci as a function of the frequency of crossing over occurring during the recombination, provided that an ordered array or sequence tagged sites along the chromosome. Genetic distance is expressed in units of crossing over or centimorgan. 1 centimorgan equals to 1% crossover and contains approximately 1000 kilobases, that is, two loci which show 1% recombination are 1 centimorgan apart on a genetic map. In a linkage, a new locus is assigned based on an already known locus. It shows that a reference locus is a prerequisite for the purpose. One map unit is defined as the linkage distance that yields 1% recombination, which means that if there is 50% recombinance or the 50% recombination has been shown, it means that the distance between the two genes must be 50 centimorgan. When a large number of genes are mapped in any given species, the genes are observed to occur in linkage groups, with one linkage group corresponding to each pair of chromosomes. Then comes the FISH, that is fluorescent in situ hybridization. It is a method that is used in gene mapping. It is very highly effective, rapid technique for the use in gene mapping. Probes are labeled with fluorochrome dyes which fluoresce in different colors when excited with UV light, that is ultraviolet light. The location of the probes are visualized under epifluorescence microscope. It is a very specialized microscope specially built only for this type of method. Chromosome painting is another method. It is one of the application of FISH techniques. It is the direct visualization of specific chromosomes by fluorescent detection by hybridized labeled whole chromosome probes is called chromosomes painting, in which the whole of the chromosome acts as a probe used to improve the accuracy of cytogenetic studies, closing the gap between cytogenetic and molecular analysis. So chromosome painting is a very important link between the cytogenetic and molecular studies. Here the whole chromosome specific probes are called paints. It is used in identification of species specific chromosomes among somatic cell hybrids. It is also used in detection of chromosomal rearrangements or abnormalities for which no locus specific probes are available. Thank you so much.